Welcome everyone. I would like to welcome Professor Richard Kelley for the new families in our group. Professor Kelley is a professor at Johns Hopkins University and was a director of the Kennedy Krieger Institute for many years.
Just a laboratory or just genetics at Kennedy Krieger.
Okay, genetics, that's our field, yeah. And he's the author of almost 200 publications. Am I right, Dr. Kelley? 175?
Yes.
You know, I'm very happy tonight because I can see names of Cat6b families since now we joined the Cat6a and B, and Dr. Kelley now also is following Cat6b patients. So I'm very excited to hear what you have to say about Cat6b, because, you know, we always spoke and, you know, learned about Cat6a, but now we have Cat6b. Is it different, the same? Should we use the same supplements?
Yes, I don't have a lot to say on that, but I do have a spot to make a few comments.
Okay.
So, so this is a brief summary of what I'm going to go through. I hope that more than half of this will be question and answer. I think that's where most of what you can learn will come from, and me too. And I shall review some of there are some old slides for those of you who have been on these talks before, just to illustrate what Cat6a and Cat6b are. And then I'll talk about our early treatment studies. We've now been treating it for 3 or 4 years in different ways. And now we're branching out into other histone acylation syndromes. The general term is acylation, which means attaching a fatty acid type molecule to a protein. In this case, and Cat6a and Cat6b are predominantly acetylation, putting a vinegar unit onto a protein, acetyl-CoA. But it also is known now to function to put other groups on the histones, modifying the interaction of histones with DNA, with the genes, and therefore modifying the function. It's extremely complex. The complexity, there are simple ways of modifying the treatments or modifying the activity in these disorders. Will be very complicated to balance all the different ways that we now know that these enzymes can modify histones. I'm going to touch upon.
Elliot.
You would have to do this in here. Can you use your own computer?
Honey, where is it?
Did someone ask me a question? Okay.
No.
And then, so I'll touch upon, I've been involved in the treatment of Rubinstein-Tavy syndrome, which is related to Cat6a. And we also are we have identified what we believe are 2 children with gain of function Cat6a mutations, which I shall describe as well. I have to use for some reason I can't, the keys are not working, but I'll use the mouse. So this is what Cat6a and Cat6b are proteins that they are enzymes, and they attach acetyl groups and, as I said, other fatty acid type groups to proteins, and that to add those moieties to proteins, it modifies the interaction when it's a histone between the histone and DNA, where histone is a protein bound to DNA that interacts with the gene regulatory sites and turns genes on and off. Histones overall are a structural component of chromatin, of the chromatins that one can see under the microscope. But they also are modified in certain ways that cause them to sort of expose the areas of the genome that can be transcribed. In the old days, we could see under the microscope what was called heterochromatin and euchromatin. The euchromatin we now know is the chromatin that is more, or the DNA that is more exposed and more functional, and the exposure of those proteins is dependent on, or the exposure of those genes is dependent on the activity of these enzymes. This is just to show that there are other proteins involved in this complex, the so-called Mos-Morph complex, and these are also involved in telling where these, in directing these enzymes to specific sites. There are even more proteins than this in the system. What it does, for those who have a little bit of experience with biology, transcription regulation is turning genes on and off. The acetylation, because these small chemicals, acetic acid, propionic acid, because they are very dynamic metabolites in the cell, the level of these compounds changes up and down depending on the various states of the cell, health or illness, or the required nutritional changes. And so that's what transcription regulation is, response to cellular stress. So when the cell is stressed, these small molecules will go to the nucleus and modify, and allow, these enzymes will modify those histones and cause these different effects. Signal transduction simply means sending through a pathway different signals to turn genes on and off, or to turn enzyme processes on and off. And then the histone regulation, or the histone modification, also has a role in basic processes like cell cycle, cells being able to duplicate themselves, and the general organization of the chromatin. These are the various systems that have been identified as more involved. So these functions affect these systems more in Cat6A than in other syndromes. So blood cells, the bone marrow, and this has been, there've been some striking examples of bone marrow failure, or nearly failure of the bone marrow, that have responded to treatment. Of course, the development of the brain is involved before birth, as is the skeleton. Heart development has been involved, and there are some what geneticists call dysmorphic features that are also a result, that they're different in children with Cat6A and Cat6B as a result of dysregulation of these systems. Just to return. Nope. I have to use my mouse, which is a little erratic. There we go. This just shows you that the Cat6A, Cat6B system is part of a very large histone modification complex involving histone methylation, which is the other primary domain, or the primary type of modification of histones. And so these complexes run up and down the chromosome, or they're available to the chromosome, and can modify, depending on the changes in the activity of these enzymes, that will modify or not modify parts of the chromosome. This is one of the things that the Cat6A came to my attention actually before it was defined as a syndrome, and a child who was followed by Dr. Rosenbaum in DC had signs of mitochondrial dysfunction, certain changes in amino acids that looked like the mitochondria weren't working quite right. And he contacted me because I treat these disorders, and I recommended a standard mitochondrial treatment, and it made a big difference. that mitochondrial treatment involves a lot of different compounds, antioxidants and some B vitamins, for example. So it's hard to know what it was that made the big difference. But I think it was Dr. Esper who contacted me next saying that her son has Cat6A and she wants the same thing that the other child had. And that was the first thing I knew, that this child I had helped treat turned out to have Cat6A. Dr. Rosenbaum was on the first paper that described the syndrome. So that got me curious, of course, into trying to figure out what exactly worked. And it became clear with a few more patients or a few more children with Cat6A that carnitine was a major factor. Just carnitine by itself can cause clinical changes, alertness, visual regard within 24 to 48 hours, which is not unprecedented in other mitochondrial disorders that are sensitive to carnitine. But it was quite clear that it was at least the carnitine that was helping the children. And this shows you, for those interested in the biochemistry, what we think carnitine is doing. It's hard to prove these things. There's lots of circumstantial evidence that this is correct, although it is hard to prove. So the Cat6A is here inside the nucleus, and it is the histone acetyltransferase. HAT is the activity that it has to put the acetyl groups onto the histone molecules represented as these balls. The question is, where is the acetyl-CoA, which is the immediate precursor or the immediate substrate for this enzyme to put the acetyl group on histones? Where is that coming from? And it comes from acetate that is transported into the nucleus. At least that's one of the sources. And this just shows how acetyl-CoA is made in the mitochondria. It gets out of the mitochondria by binding to carnitine, and that is converted back into acetyl-CoA outside the mitochondria, inside the cytoplasmic space. That is converted to acetate, and that penetrates the nucleus. So it certainly made sense that by giving carnitine, it would increase the ability of acetyl-CoA to get out of the mitochondria and into the nucleus. The problem with Cat6A is that there is only one of the two Cat6A proteins that every person has. There are two genes that encode usually the same Cat6A protein in children with Cat6A syndrome, and similarly with Cat6B. One of the genes is abnormal and doesn't encode a normal protein. So there's, in general, 50% of the enzyme activity of Cat6A, our model here, and in general, working with enzyme deficiencies, which is what biochemical geneticists do, what metabolic specialists do, to make a weak enzyme work better, simply flooding that enzyme or giving that enzyme a lot of what it needs to work, in this case acetyl-CoA, can increase the activity from a baseline of 50% to maybe 60 or 70%, which can really make a big difference the way these systems work. It doesn't have to be 100%. So we think that the carnitine is facilitating the production of acetyl-CoA, which strengthens or increases the residual activity of the Cat6A, which is only without treatment 50%. However, it's a little more complicated than that, as it usually is, because fairly recently, it was found that there is an enzyme that is in mitochondria that is called pyruvate dehydrogenase, which converts pyruvate, which is a derivative of glucose, into the same compound, acetyl-CoA. And we have known for decades that carnitine is an activator of this enzyme. This enzyme is peculiar in that it exists, not peculiar, it's one of many enzymes that exists in an active and inactive state. And carnitine increases or converts more of the enzyme from the inactive to the active state. So this is another way that carnitine could be working. It could be increasing the activity of the pyruvate dehydrogenase, which occurs in the nucleus. It's the same protein that's in the mitochondria, it just gets into the nucleus as well. And then that would increase, if by giving carnitine would increase the activity of that, increase the acetyl-CoA, and therefore increase acetylation of histones. So by one mechanism or the other, carnitine makes a big difference. This just shows you where this goes back into the mitochondria, showing that acetyl-CoA comes out. The citric acid cycle is where energy is made inside the mitochondria, and this just shows you for where acetyl-CoA comes from. I put this, and it also shows you that amino acids are a very good source of energy for mitochondria. And they're in a number of mitochondrial syndromes, we need to give extra amino acids because when mitochondria are weakened in a variety of ways, they often resort to using amino acids like methionine as a source of extra energy. The problem, however, is that there's not much methionine around, or it's the least abundant, one of the least abundant of the amino acids. And therefore, if you start using methionine for just energy metabolism, then it's there's less around for doing other important functions of methionine other than making new protein. And that's what we have found is the cause of the bone marrow failure in CAT6A, as we have seen methionine deficiency as a cause of bone marrow problems in other mitochondrial disorders. I also show this diagram here to indicate that acetyl-CoA is all, or one of the things we'll get to later. Citrate, we have found that citrate level is low in mitochondria, in CAT6A syndrome in the blood. Citrate is made in mitochondria, part of the what's called the citric acid cycle, and it is then exported out of the mitochondria for a variety of purposes other than energy metabolism. However, when mitochondria are impaired and are not able to make enough citrate for running the citric acid cycle, the mitochondria get very stingy and they don't send citrate outside the mitochondria and for other purposes, one of which is to make, so citrate is converted into acetyl-CoA. So again, that's a reason for enhancing the acetyl-CoA movement out of mitochondria with carnitine. But acetyl-CoA is a source, acetyl-CoA combines with a vitamin-like compound called choline to make acetylcholine. Acetylcholine is the major neurotransmitter that powers the gut musculature. And to make a long story short, we found that almost all children with CAT6A have low levels, quite below normal levels of citrate in the blood, and the blood correlates with the amount of citrate getting out of the mitochondria. And there really is no other way to make citrate except by the mitochondria. And therefore acetylcholine, which is very important, of course, for making the gut move, that becomes deficient, which is the cause of all of the intestinal dysmotility, severe colicky pain, and other GI symptoms and problems that are seen in CAT6A. And in general, supplying citrate as a medication to not need the mitochondria to provide citrate, giving citrate increases acetyl-CoA in the cytoplasm and acetylcholine. And this is actually a treatment I've been using for about 30 years in other mitochondrial syndromes. And it just has turned out that children with CAT6A, and I don't have as much data for CAT6B, but I would expect it to be similar. I don't know that it is similar yet, but it might be. Giving citrate as a medication generates or reverses all of these GI problems in most children who take it. And it's not, compared to what we often use citrate for, it's really not a huge amount of citrate, and it's well tolerated by most children. I think I can skip this because I've gone over most of these points before. So this is just where we were in 2018 when we pulled together our first set of data on 26 children with CAT6A. And what we established was that in addition to the low citrate and unrelated or as a the citrate we now understand is probably a major cause for the deficiency, by boosting the citrate, as I explained, we can make the gut work better. But there are other mitochondrial abnormalities besides the low production of citrate that, as I explained, can be the cause of bone marrow failure. And we have to look at each child individually to determine if they have these other problems. And only a minority of young children with CAT6A have these other mitochondrial problems that require additional treatments, such as antioxidant cocktail, coenzyme Q, vitamin C, vitamin E, and amino acid supplements, as I explained, bone marrow failure. So each is what we have seen evidence of is that the mitochondrial dysfunction in some children with CAT6A who have had certain laboratory tests going back many years, it does appear that there can be increasing mitochondrial dysfunction, so that some of the adolescents and young adults with CAT6A have more of the characteristics that we see in classical mitochondrial diseases caused by a primary mutation in a mitochondrial gene. And fortunately, they do respond to the same kind of treatment that we worked out for those other mitochondrial diseases. My role right now is largely looking at looking at the laboratory results and the clinical problems of each child and determining what the best treatment is. There is, I can say that probably it seems to be that carnitine, and we also encourage the use of a B vitamin supplement to help the carnitine work better. That carnitine appears to be universally required for the typical CAT6A mutation and presumably CAT6B as well. But the rest is individuated, or we have to decide for each child after doing a series of metabolic tests what the best treatment would be. Unfortunately, because it's hard to pull all this kind of information together in an adequate publication that satisfies all the requirements of publication, especially when one is treating patients individually rather than part of a protocol. These things have not been published except as one abstract, and I'm sure some parents listening have had trouble getting their physicians to believe this. And unfortunately, not all physicians will contact me to sort of get to learn more about things. And it's unfortunately, it's caused some upset among geneticists that they're being asked to see children with what the literature says is not a mitochondrial disorder. Pediatricians send the children to the local metabolic specialist to look for their mitochondrial problems, and they don't find anything because it's subtle. So I just mentioned that it's a problem, and it sometimes requires just finding the right kind of specialist who is willing to act on information before it is published. This just to give you a sense of the complexity. These are compounds like acetate. It says 2 carbons, 4 carbons, 3 carbons, 4 carbons with an unsaturated bond, and so on. And these are all the different. The K means a lysine residue in the histone, and that's where these groups will attach, certain lysine groups. And this just shows you the variety of substitution that can occur. Acetylation is the predominant one at this residue, lysine 44. But all these other ones, propionate in particular, we've been interested in, as you see here, a large amount of propionate is attached to this particular one. And this is what I'm saying, it's very complex to try to sort all these, to figure out how to replace all these different functions of a deficient enzyme. We can work one compound at a time in terms of research, but what will be an ultimate ideal treatment is left to be determined. This is just to show that we've been considering the propionation, which is known to be a substrate. Propionate is the substrate of the CAT6A enzyme. This is a study just showing that there is abnormal, or there's deficient propionylation, which means adding the propionate group, this 3-carbon group, to histones is impaired. H3 is one of the histones, is impaired in various developmental disorders. Just to briefly review, we had considered and are still thinking about using triheptanoin, which is now being used to treat another group of metabolic disorders, disorders of fatty acid metabolism. And the idea here is triheptanoin, which is a 7-carbon fatty acid, instead of being like olive oil, oleic acid, triglyceride, it's a C7 compound, triheptanoin, and that is catabolized, broken down in the mitochondria to acetic acid and propionate. So it's one of the, it's a medication or a supplement that can be given that should generate extra propionic acid. In the syndromes that are treated with this propionic acid, they want this to be converted into succinate, which it is, but it must go through propionate to begin with. But what I found in testing some children who are taking triheptanoin, you don't have to know this, that the most abundant acyl group in the blood is not C3, but it's C5. So the first step in the catabolism, in the metabolism of heptanoic acid is to break it down into pentanoic acid, 5 carbons, and that's what's most abundant. So it's not clear that triheptanoin is going to work in treating CAT6A, but it is still a possibility to be looked into. Many of these types of, or this type of research is best done in an animal model where you can more easily determine exactly what is happening in the metabolism of heptanoic acid. So there are a number of things, discussions and research going on, and amino acid metabolism is, we still don't understand why asparagine and citrulline levels are high in CAT6A. What it tells us that mitochondria are not working right. But exactly what is in mitochondrial dysfunction is causing that amino acid abnormality is not clear. And as I said, it's better to study treatments, possible treatments in the mice. We had hoped to do a protocol this year, combining some of these ideas for treatment, but the pandemic derailed it both in 2020 and 2021, possibly next year. And sorry, I don't, my mouse is not working right. So one of the possibilities is to look at triheptanoin. And then the other thing that we are doing as this story gets around to other disorders is beginning to look at other syndromes with similar function, with similar defects. And one of those is a disorder that I have seen myself a number of times in years past called Rubinstein-Taybi syndrome. And it is different from CAT6A, but this is just to show that the two proteins that have been shown to cause a deficiency of these proteins causes Rubinstein-Taybi syndrome, and these are histone acetylases with structures that are not unlike those of CAT6A and CAT6B. Here's the histone acetyltransferase enzyme region right here. And it's just a diagram to show you that these two proteins are both involved in putting acetyl groups onto histones, illustrated here. This is Rubinstein-Taybi syndrome. It has a number of distinctive features. They have broad thumbs in general. It's more the articulation of the thumb is abnormal. It flexes in odd ways. They often they have somewhat small skulls, small heads. They have microcephaly as in CAT6A. They have a beak nose sometimes. And here's the broad toes that are characteristic of Rubinstein-Taybi. This also shows the abnormal articulation and underdevelopment of the thumb. So it's different than CAT6A and CAT6B, but it does involve the skeleton, and children with Rubinstein-Taybi can also have heart problems. And then the last topic to mention is that two of the patients, I think one's an adolescent, one's a younger child, probably have what's called a gain of function mutation in CAT6A. This is just giving the regions of the protein, of the CAT6A protein, which in the CAT6B is almost the same as this. There are different regions of the protein that interact with the histones in different ways. This is the enzyme part, but a mutation in almost any portion of the gene can destroy the activity of the gene and of the protein encoded by that gene. And this just lists all the different ones that have been found. When most of these proteins eliminate the activity, as I said, of one of the two, or the protein made by one of the two genes. One of the genes is defective, and the protein made from that, the CAT6A protein made from that defective gene doesn't work. So all the work is dependent on the normal gene. These are almost always new mutations. It's different in CAT6B. Some of the mutations can be inherited, so-called hypomorphic ones. And the two mutations that we have identified as possible gain of function, pretty likely gain of function. One is here. A mutation in this region is more likely than other mutations to cause a persistence of the protein. And without going into the details, if you have an abnormal protein hanging around, sometimes it can by abnormal interaction with its pair, with its other proteins, it can actually increase the enzyme activity, increase the acetylation activity, and one of the mutations that we believe the acetylation activity is increased is a so-called splice site mutation that affects this region of the gene. The genetic details are not as important as the clinical findings, because what made us think that we had a gain of function mutation is the children have developmental delays, not greatly different from CAT6A, but they, instead of having high asparagine and citrulline levels, they had low asparagine and citrulline levels. And similarly, they had high citrate, these two children, rather than low citrate levels. They also had no significant history of intestinal dysmotility. And so it becomes a question of, you know, what can we do to, is this a treatable syndrome, and how would we treat it? And this is the last slide I was working on, obviously. And one of the possibilities I was going to put here is valproic acid. Valproic acid, which is used as a seizure medication, is no, I'm sorry, I have it backwards. One of the possibilities for treating CAT6A is with valproic acid, which is a deacetylase inhibitor. So it inhibits the enzyme that takes off the acetyl group. And so what we have to find is an acetylase inhibitor of some sort, in theory, to reduce the activity of the acetylation. What appears to be increased rate of acetylation causing the opposite biochemical phenotype. And that's a complicated, it's really not clear how to go about doing that, because there's been no precedent in CAT6A for gain of function mutation. There is a syndrome, a subset of the CAT6B, which is known to be a gain of function mutation. And therefore we might learn more from that syndrome, the CAT6B equivalent, which has maybe 15 or 20% of individuals with CAT6B mutations have gain of function mutations. So apologize for the incomplete slide, but I think that is basically it. And time for questions.